Okay, can you hear me?
Yeah, I had to log off and redo my browser for some reason. Can you hear me?
Okay, it's better.
Okay, you
Okay,
can
let's
hear
start.
me. Can you
Sorry.
hear me? Can you hear me?
So it's my great pleasure to introduce Professor Susan Lunti. Professor Susan Lunti is the Ralph Adams Distinguished Professor of Chemistry and Pharmaceutical Chemistry at the University of Kansas. And Susan got his PhD in analytical chemistry in Purdue University. And she has a distinguished curriculum, if I can say this, because I have to touch here. She's appeared on the top 100 more influential researchers in analytical science in the magazine, The Analytical Scientist. So Susan is a really a known person in the community of analytical chemistry with huge contributions. If you see the CV, is really difficult to get some points to put here, but just some numbers. She has 171 published papers in the field of novel and electrochemical detection methods for capillary electrophoresis, detection methods for microchip electrophoresis, online microdialysis coupled to capillary and microchip electrophoresis, electrochemical methods for detection of reactive oxygen and nitrogen species. She has 22 book chapters, one book published, 4 patents, um, 57 theses and dissertation supervisions, and 289 presentations. This will be the 290 90 presentation. So Susan stay here for me, could be here at many times. She's also from the advisory board of this institute, so international consultant for this institute. So it's a great pleasure, Susan, to have you here. Um, it will be better if you present in the presence, but we are really happy to have you here, even in a remote mode presentation. So thank you so much for giving us this time to present your work here for us. So it's up to you now. Thank you.
Yeah, thank you, Fracassi, for the really nice introduction. And I will be out there in November for LACE, so I'm looking forward to seeing everybody at that time. And I also have a couple of students, one from Fracassi's group and one from Wendell's group, coming at this weekend. So I'm looking forward to having a couple of Brazilian students in my group this year. So what I thought I'd talk about today is just sort of an overview of what our group does in the area of bioanalytical applications of microchip electrophoresis. Uh-oh, let me see if I can do this. Okay, so I'm going to talk about sort of 4 applications: separation and detection of reactive nitrogen and oxygen species. And Professor Fracassi has been involved in this work over the years. Some work we're doing on looking at RNOS biomarkers for clinical applications, single cell analysis systems. Jay Sibitz has joined my lab and has been working on that, and then on animal sensing. So one of the overriding areas that we're interested in my group is neuroinflammation. So how neurodegeneration happens. And we're really interested in sort of the smaller species that are involved in neurodegeneration, such as nitric oxide, peroxynitrite, and also the compounds that can prevent neurodegeneration, like glutathione and ascorbic acid. And so we've been trying to develop methods to look at both these pro-oxidants, nitric oxide, superoxide, and also the antioxidants, which can help prevent neurodegeneration or inflammation. So our take on this is to use a separations-based approach. So there's lots of sensors, single analyte sensors out there, but our idea is to use microchip electrophoresis to separate out the different components and then detect them with a selective detector. So that way we can measure multiple analytes simultaneously, and it also eliminates any interferences. If you have a sensor for just one particular analyte, there may be interferences that could give you an improper result. So on the left is some of the methods that we've used. I'm going to mainly talk about electrochemical detection and LIF detection in this talk, along with the analysis systems. And then the analytes that we've been interested in, especially related to oxidative and nitrosative stress, are all electrochemically active. So the nice thing about electrochemical detection for microchip electrophoresis is that reactive oxygen and nitrogen species are by definition oxidants, and so most of them are electrochemically active, and most of the antioxidants are also electrochemically active. So electrochemistry is a really nice approach for looking at reactive oxygen and nitrogen species. So I think probably most of you are familiar with microchip electrophoresis, but I just wanted to go over it just a little bit. So the simplest microchip is just a simple T. We use voltages. We apply voltages across the channel. It's similar to capillary electrophoresis, except that the chip's maybe 5 to 15 centimeters long, and we can do the separations in this very small diameter channel. We can do very fast, efficient separations. The separations are based on charge to mass or charge to molecular volume. And so it's really ideal for separating both large and small charged analytes. So this just sort of summarizes some of the advantages. Like I said, we can separate monitor multiple species simultaneously. We can do really fast separation times because the channel is so short, and the voltage that we apply over the channels, the field is usually 200 to 500 volts per centimeter. So we can get very fast separations, generally under a minute, sometimes a couple minutes. Also, this has a really small footprint. So for some of the applications that I'm talking about, the portability is important. We can use multiple types of detectors. We can use electrochemistry. I know groups in Brazil have used conductivity a lot. We can also do laser-induced fluorescence. And I think also key with the microchip format is it's easy to integrate sample prep or other steps into the chip. So I will talk today about some of the single cell analysis and in vivo monitoring using microdialysis. So first I'll start with electrochemical detection. I think actually my first interactions with Brazilian scientists, Fracassi and Wendell, was a really long time ago when they were working on electrochemical detection, and I was at a LACE meeting. And electrochemical detection, I feel, and I think they do too, has a lot of advantages for the microfluidic format and the microchip format. The electrodes are planar, so they fit really well into the chip format. It's easy to integrate the electrode on the chip. As I mentioned, they're selective for redox active analytes. So in blood and other tissues, there aren't that many things that are redox active. You can use a variety of electrode materials, including modified electrodes. You can do multiple electrode configurations. You can use, I won't talk about this, but if you run 2 different voltages, you can identify a compound based on its voltammetry, similar to running a UV spectra. And also you can miniaturize the potentiostat and associated electronics to make a portable analysis system. So first thing I'm going to talk about is sort of just RONS in general, how we detect those species, and also some of the products of reactive nitrogen and oxygen species with proteins and peptides. So nitrated proteins have been shown to be present in high concentrations in the brains of Alzheimer's patients. And a group in Kansas City has shown that nitrosyracine containing proteins and peptides in blood might be a potential indicator of Alzheimer's disease. And then there's other modifications of proteins that can be due to superoxide or hydroxy radical that's present in the system. So here, this again is just some of the electroactive analytes. So actually, I got a few slides from the past when Fracassi was here on sabbatical just to show some old work, but One of the first things we wanted to do is just see if we could detect nitric oxide and peroxynitrite. So nitric oxide reacts with superoxide to form peroxynitrite in vivo, which can then lead to protein oxidation and nitration, and as well as lipid peroxidation and DNA oxidation. So this is actually Fracasse's work from quite a while ago, where he was actually able to take proline nanoate and detect nitric oxide that was generated from the proline nanoate. But as you can see, we also detect the proline nanoate. So this separations-based approach allowed us to separate nitrite, proline nanoate, and NO using the microchip electrophoresis. So if we were just doing a single sensor, we'd detect all three of these compounds. We were also able to stabilize and detect peroxynitrite. Peroxynitrite is not very stable at pH, at physiological pH, at pH 7, but because electrophoretic separations don't have a stationary phase, you can use really basic buffers to run your electrophoresis in. And so we did this separation and reverse polarity at a high pH, and we were able to detect the peroxynitrite. So we, Dula and Gunasekera, my group, developed a sort of a separation of a bunch of oxidants and antioxidants here. So you can see nitrite, which is a indicator of nitric oxide stress, glutathione and ascorbate, which are antioxidants, and then tyrosine, which I'll talk a lot more about, and then hydrogen peroxide. And so just one example of a cell study, nitric oxide is produced in macrophages. So in macrophages, if you stimulate macrophages, you trick them into thinking that there's a bacteria nearby by adding lipopolysaccharide. The bacteria will then start producing nitric oxide. And then if you add polymerisotyl acetate, it'll start producing peroxynitrite. But when we just for an example study, if we added LPS to the macrophage and we do the analysis of the cell lysates, we can see that we see the production of nitrite here. The LPS stimulated is higher than the native macrophage. So that's sort of how we got started into this. We were wanting to develop some methods to look at peroxynitrite and nitrated compounds. And so we sort of moved into looking more at protein modifications by reactive nitrogen and oxygen species. So phenylalanine and tyrosine can both react with superoxide, hydroxy radical, and peroxynitrite. And so there's a variety of amino acid modifications you can get, including meta and orthotyrosine, nitrotyrosine, L-dopa, and dityrosine. So Danushka in my lab developed a ligand exchange micellular electrokinetic chromatography method to separate out all these modified amino acids. And so this used a ligand exchange method where you have copper you have a ligand to copper. The ligand is 4-hydroxyproline, and then in the presence of these tyrosine and its derivatives, it will change the mobility of the tyrosine and make it easier to resolve the compounds. So this shows, this sort of jumping to the end result, but after he developed this separation using capillary electrophoresis, he was able to transfer this to microchip electrophoresis with electrochemical detection. So here you can see all the different modifications, L-dopa, also the 3 isomers of tyrosine, the para, meta, and orthotyrosine, which would be produced from the reaction of phenylalanine with hydroxy radical, also nitrotyrosine, and then we also, and then dityrosine, which is a sort of a diphenol. So while we were looking at these modifications, one thing we know is that if we buy nitrotyrosine or peroxynitrite from commercial vendors, it seems like it's always sort of a mess. So Kelsey Shilley in my lab has been developing or developed a method for producing peroxynitrite. And so basically you take nitrite with hydrogen peroxide in an acidic solution that forms the pernitrous acid, which then will react with your compound in under basic conditions and form peroxynitrite. And so you can see, actually, you can do this, monitor the reaction by CEUV. The peroxynitrite absorbs at 302 nanometers, while the rest of them absorb at 214 nanometers. But you can see you get nitrate, nitrite, and hydrogen peroxide in there. So one thing she did is she sort of looked at the reaction time. So she would put the nitrite and the hydride, she'd set up the nitrite reaction or the peroxynitrite reaction. And then she would add tyrosine at different intervals after making the peroxide or the pernitrous acid. And you can see that if she adds the so this is the peroxynitrite, you can see it degrades quite quickly. If she adds the tyrosine right away, she gets a pretty good yield of nitrotyrosine. But if she delays adding the tyrosine, then she starts getting just the tyrosine back. And you can see that from this these separations that she has. So if she lets the peroxynitrous acid degrade, all she gets back is tyrosine. If she adds the tyrosine immediately, then she gets actually not only nitrotyrosine, but another peak, which we've later discovered to be dinitrotyrosine. And then these are just the standards of the two compounds. So looking at this, we're trying to figure out, well, can we selectively detect, one of the things we wanted to do is see if we could selectively detect nitrotyrosine derivatives or nitrotyrosine compounds in the presence of tyrosine. So if you use oxidative microchip electrophoresis with electrochemical detection, you can detect tyrosine, nitrotyrosine, and dinitrotyrosine, because the nitrotyrosines are both, can be oxidized and reduced. So if you look at the three compounds here, tyrosine, nitrotyrosine, and dinitrotyrosine, with oxidative detection, you get all three compounds. But if we go to reductive electrochemical detection, where we apply a reducing potential at the working electrode, then we can be selective for nitrotyrosine derivatives over the tyrosine derivatives. And so why is this important? Well, if we want to look for this in a biological sample, and nitrotyrosine is a modification of just some proteins, there's going to be a lot less nitrotyrosine in there than tyrosine. So if we do a enzymatic digest or a HCl digestion, we're going to get a huge peak for tyrosine and really small peaks for nitrotyrosine. So the chances are we won't be able to see the nitrotyrosine in the oxidative mode because it'll be swamped out by the tyrosine peak. So here with reductive electrochemical detection, we see that we can get some selectivity for the nitrotyrosine. So why are we interested in this? Well, Christian Schoeneich and Russ Swedlow at the Alzheimer's Center, they're interested in looking at nitrotyrosine in mitochondrial proteins that isolated from the white blood cells of patients. So they've actually they have a very long procedure they use to isolate white blood cells from Alzheimer's patients' blood, then isolate the mitochondria, and then measure the nitrated proteins in those mitochondria. So one of the things that they would really love to have is to simplify that and be able to detect these nitrated peptides in sort of more of a clinical type assay. So we're sort of working backwards. We're developing a selective method for these peptides, and then we're going to look at the nitrated proteins and then work back on the enrichment steps, which could conceivably all be done on a chip. So the model system we've used is this protein called phospholambin. It's the one that Christian Schoeneich's group had used to do some of the initial studies. It's a 52 amino acid peptide. And in particular, it has this one peptide, QYL, that would be nitrated with peroxynitrite. And so Kelsey Schilly and Riley Stegmeyer in my lab developed a separation of these peptides of interest. So actually, this is the AYL. There was another protein that they had that had this F-salar. So we're basically separating out the nitrated and the non-nitrated compounds in this series of things. So this again is another just a summary slide. So they developed a nice CEUV separation. It's really interesting because all the nitrated peptides are well separated from their parent compounds. They're all much more, they have much more negative electrophoretic mobilities, as you can see. And so they were able to separate out tyrosine, all the native peptides, which come out first, and then all the nitrated peptides. When we did MEEC in the oxidative mode, we were definitely able to separate tyrosine and nitrotyrosine, and we're able to get some of these peptides. But we're still working on that, and I think one of Fracasse's students might be picking this up in a week or so. But if we do reductive electrochemical detection, this is something that Indica did. You can see that we can separate out these nitrated peptides, and we can selectively detect them. And this is using microchip electrophoresis, so you can see it's really, like a minute and a half, although the separation itself's about a minute. So this just, we're working on this. This is something we're really interested in doing, is looking at these nitrated peptides. So the idea next is to take the peroxynitrite that we synthesize in-house, react it with phospholamban or other proteins, then enzymatically digest those and see if we can selectively detect the nitrated peptides. Also, we're looking at trying to just determine total nitrotyrosine by doing some sort of chemical digestion. And then eventually we want to get the mitochondrial proteins from Fedlow's lab that they isolated from the blood and analyze those. And then, of course, the ultimate goal is to develop an integrated system. So the next thing I wanted to talk about was cell analysis using microchip electrophoresis with fluorescence detection. So here we're using a bunch of different dyes, standard dyes that are used by a lot of imaging people. So we use DAF-FM for nitric oxide, Mitosox for superoxide, although there's a new dye that Jay's looking at. And then we have also, we can look at total RNOS, and we've also tried to do some work with glutathione. And this is Jay Sivitz, who's working, and then Giuseppe Caruso. So with the, I'll just give the example for nitric oxide here. We are trying to look at how much nitric oxide is produced in a cell. And so we're adding both DAF-FM diacetate and 6-CFDA to the cell sample. So those macrophages that I talked about at the very beginning. So when we add the dyes to the cell, they pass through the membrane, and there's an esterase that brings them, they're brought into the cell. And then when the esterase acts on the dye, it traps the dye in the cell, and then then in the case of the DAF-FM, it reacts with in the cell to form this DAF-FMT. And the CFDA just reacts with the esterase and is trapped in the cell, but that gives us a good idea of the volume of the cell and the activity of the esterases, so it can sort of be used as an internal standard. So a long time ago, our initial thing was we just did it in a bulk cell lysate. So we incubated some macrophages with the DAF-FM and the CFDA, and then we just separated out the two compounds and measured their fluorescence. So the interesting thing is, and why we feel like we need separations, is the DAF-FM also reacts with degradation product of ascorbic acid. So you can get an enhanced signal for DAF-FMT if you're just doing imaging compared to doing a separation and actually measuring the peak area. So here you can see this is just the calculated amount of nitric oxide if we using this approach using the CF as an internal standard, and you can see we see an increase in the amount of NO that's produced. So then we were wondering, well, is it so macrophages have many different states and many different they have M1 and M2, and they're stimulated in different ways. So we wanted to see if all the if the macrophages were if we did single cell analysis, would it be similar to this bulk cell analysis? So Chris Culbertson's lab has this single cell analysis system that we've been using, that now Jay has moved to our lab. And in this system, you pump the cells one by one into the chip, where the electric field, there's an electric field that's set up for right here for the separation. So as the cells go into the intersection here, the electric field that's used for the separation lyses the cells, and then the cell lysate is separated in the channel. And this shows a little better picture where the cells are coming into the chip, and here one by one marching into the chip. Then they get to the part where the electrophoretic field is applied, and then they're lysed in the channel. And then we're getting the electrophoretic separation of the analytes in the channel. But since we're only looking for the fluorescent dyes, we're just seeing the fluorescent dye here, and then we can separate those out. So Chris has got this going at 15 cells per minute, so you can do really high throughput analysis. And this just shows, so actually, because the lysis is so fast, some of the CFDA, you actually get 2 peaks because one ester group versus 2 off. But you can see the DAF-FMT here is apparent. So this is one single cell, this is another single cell, another single cell. So when we did this, you can definitely see if you take 100 cells and you look at the ratio of the DAF-FMT to the 6CF, the stimulated definitely are producing more nitric oxide, but you can see it's also a much broader distribution than it is for the native peptide. So here this cell is very different from this cell. So we're interested in seeing what's the difference in these different cells. Well, here, most of them are in this middle part here. And when we look at, if we just compare like the average that we get from the bulk cell analysis with what we get when we take all the single cell analysis results together, they come out pretty similar. So if you did bulk cell analysis, you'd get an average, but you wouldn't see this sort of distribution of these cells. So Jay's moved on with this, and he's looking at microglia. So microglia are the macrophages in the brain. So we're really interested in those because they can cause all sorts of neurodegeneration and oxidative stress in the brain. And so he's using this SIM-A9 microglia. And here you can see he's got the separation of the native and stimulated, and then you have the interference here as well. So what Jay saw actually was for the microglia, it really wasn't a Gaussian distribution. It was more of a log normal distribution. And so we were really interested in sort of finding out what's happening here. And so he's working with our collaborator from Italy to look at, what the effects of different stimulants are on this distribution pattern, as well as some antioxidants. They're interested in particular in a natural product called carnosine. Okay, so the last part that I wanted to talk about is doing in vivo studies. So I've shown how you could potentially use the chip for a clinical application, integrating it with the microchip with sample prep for blood sampling, and also how we can do this single cell analysis. So another thing that we've been doing is trying to over the years is coupling microchip electrophoresis with microdialysis sampling. So the idea here is to make what we call separation-based sensors. So we use microdialysis sampling, which I'll explain in a minute, and then we use microchip electrophoresis to separate out the compounds of interest. And then I think all the applications I'm showing today are amperometric detection. So the microdialysis sampling acts as sort of like a generic sampling method. It's I'll show in a minute. It's like a synthetic blood vessel. So you can really analyze, you can really sample any sort of compound with the microdialysis sampling. And then we can optimize our separation and detection modes for the analytes that we're interested in. So for those of you who don't know what microdialysis sampling is, it's a really pretty simple method, actually. It's a dialysis membrane, so a very small dialysis membrane. So usually, 100 microns or so, or 250 microns across. And it can be implanted at the end of a tube for brain sampling, or in this case, it can be put in a linear probe type configuration. But the main thing is with this microdialysis sampling, this dialysis membrane, which is basically a kidney dialysis membrane, it excludes large molecules, and only small molecules can pass through this membrane. So what you do is you pump a perfusate really slowly through this probe, and then as you pump, small molecules from the extracellular fluid that you're looking at, or if it's an in vitro experiment. But anyways, the small molecules diffuse across this membrane into the dialysis membrane, and then they are pumped to the analysis system. So from an analytical chemist point of view, this is a fantastic technique because the perfusate is usually a ringer solution, basically a buffer solution that has similar ionic composition to whatever tissue you're looking at. So it's a really clean aqueous solution. And because this microdialysis membrane excludes large proteins, the samples that come out are really clean. They're just basically small molecules in a salt solution. So it's really nice for an analytical chemist. You don't have to do any centrifugation or precipitation. Another thing that's important is enzymes that will degrade peptides and proteins, or enzymes that will degrade peptides and other small molecules, are also excluded by this membrane because they're proteins. So you don't have to add enzyme inhibitors to your sample either. The only caveat is if you're doing some sort of drug study, or you're trying to look at something that's highly protein bound, then this can be a problem, because if a drug is protein bound, like this little purple thing with the green attached to it, then it will not go across the membrane because it will be attached to the protein. So this shows a linear probe. That's one of the examples I'm going to show with our on-animal system. And basically, it's just a thin membrane over a tube here that you just basically sew into the skin or a tissue, and then continuously monitor what's going on. So for brain studies, you usually use a brain probe, which has this dialysis membrane on the bottom of the probe. So you have a guide cannula that you put into the rat's brain, and then you add the brain probe in there. And then you can perfuse. So normally you'd put this in a rat in a bowl. You detach the tubing to the rat. Once the rat recovers from surgery, you can do the microdialysis sampling for long periods of time. It basically does not take any fluid from the brain. Everything's based on diffusion. So you can do continuous monitoring for hours. In fact, and some people have done it for days in our labs. So it really acts sort of like a synthetic blood vessel. Things that are in the extracellular fluid will diffuse across to the probe, and then we can monitor what those are. But we can also perfuse things into the brain and look at the metabolism at a specific site. Or some other examples are looking at, which we haven't done, but Craig did, was looking at the metabolism in tumors or versus healthy tissue, or looking at muscles, liver. There's all sorts of applications. But the one I wanted to show is obviously with microchip electrophoresis. So we developed this chip where we could connect the microdialysis sampling directly to the microchip electrophoresis. So the idea here is to be able to continuously monitor the perfusate as it's going through, as it's coming out of the microdialysis probe. And so what we've been using the gated sample introduction, so we can use the voltages that are applied to the different parts of the chip to actually introduce the sample into the chip at regular intervals. So if we have the high voltage between, like in A, between here and here, then there will get a gate where no sample will go into the chip. But if we float that for just a second, then a small amount of sample will go into the chip and they'll be analyzed. So why is this important? Well, we wanted to see if we could miniaturize this whole system and put it on an animal, and using being able to just use potentials to control the injection makes it a lot easier to do things remotely than having a on-off pneumatic pump, a pneumatic injector or something like that. So the idea was to take this giant system we had on the bench. So this is the power supply, this is the electrochemical potentiostat, the chip's up here, and then here's the pump that goes into the microdialysis probe. And this doesn't even show the animal, but the idea was to miniaturize this whole thing and put it on animal. And so we did this quite a while ago, but we decided to use a sheep first. Now you might say, why sheep? Especially, I think Brazil, you have a lot of cows in Brazil. Well, we have a lot of cows here in Kansas, but sheep turn out to be, well, first of all, I'll show you the system is not small enough yet to put on a rat to look at rat studies. And sheep are pretty docile animals. They tend to not be they would tend to wear it easier than pigs or cows. And they also, there's a lot of interest in social behavior and depression with sheep. So this shows the overall system. So initially we didn't do any brain analysis. I want to say that if any of you do rock climbing in any of the mountains, we actually used a rock climbing harness that they use for dogs to put the thing on the sheep. So here's the system on the sheep. So here's the microdialysis pump. It's just a battery powered syringe pump. The Pinnacle made the electronics, so the whole thing's run right now by a laptop battery, but it could be a lot smaller battery. But since it was a sheep, we had some more real estate. And then we can put the chip right here and run. The whole thing is run by Bluetooth, so by a computer remotely. So the sheep is not tethered at all to tethered to the analysis system. It can be freely roaming. And then we'd put the microdialysis probe underneath the skin. So we wanted to look at transdermal delivery. So we didn't do any brain chemistry yet, but we picked an analyte that we were pretty familiar with, the nitrite. So we took nitroglycerin, which is used for heart attacks and cardiovascular disease. And we put nitroglycerin through the skin, and then we could look at the production of nitrite from the nitroglycerin. So nitroglycerin produces nitric oxide, and then you'll end up generating nitrite. So when we did that, we were able to monitor nitrite like every 10 or 15 seconds continuously. There was a lag time for the because we're pumping at the slower the flow rate, the better the recovery. So we're pumping at a pretty low flow rate. So there is a lag time, but then you can see every about 10 seconds, we're able to get data on the amount of nitrite that's being produced. And we did put a second probe in the sheep on the other side. So they weren't exactly the same sample, but we just sort of wanted to look at if the perfusion kinetics were about the same. And so what's in blue is what would be done with HPLC. So you have 10-minute samples of that you're analyzing the nitrite. And so this is on the other side of the sheep, and the red is what we were getting from the online microdialysis system. So that showed us that we could actually set the system up and we could put it on a freely roaming animal and make it remote. But the really important things that people are interested in are catecholamines and other neurotransmitters, as well as indicators of oxidative stress and metabolism deficiencies. So I've had several people in my group working on developing separations of the catecholamines to couple to the online microdialysis system to monitor, continuously monitor neurotransmitters. And the advantage of this over, say, in vivo voltammetry is with in vivo voltammetry, you really only measure dopamine, while here you can measure multiple compounds simultaneously. So here, I thought I had the actual separation. I might have deleted it, but we want to monitor dopamine release. So here we had the probe in the brain of the rat, and we had a perfusate here. First, it was just phosphate at pH 7.4, but then we changed it to a high potassium solution. And you can see sort of here that there's a increase in dopamine in the high potassium perfusate. I think, oh, here, you can see it better. So here's electropherograms for the first stimulation, the second stimulation, and then the last 10 minutes of the second stimulation. So if you plot this out, what you can see is that we can definitely detect the dopamine release. And we only looked at dopamine in this experiment, but even though they were all separated, but we're still working on trying to get limits of detection so we can detect endogenous dopamine. So we were able to detect changes in dopamine, but not the endogenous level. So we've been trying to work on improving the LODs for our catecholamines so that we can continuously monitor them from basal levels to increases. So there, the ultimate goal is really correlating neurochemistry with behavior. So one of the things that we'd really like to do is, and we did a little bit of this with Pinnacle. If you go to the paper on this, we actually, I think, have a picture of the sheep going right to the camera and licking it. But let's say you had, what is it, Rio versus Sao Paulo soccer sheep. And you had these three sheep are Rio de Janeiro fans. These two are, what is it? What's the name of the team again? I have a bunch of the shirts. Anyways, Sao Paulo team. And then what you could have is if you had numbers on the sheep, or then you could continuously monitor by having cameras in different parts of the pen. If you had different cameras, you could monitor their behavior. And then with the telemetry system, you could correlate that with the neurotransmitter. So that's really a high-end goal of what we want to do, sort of the holy grail. But if we could monitor, for example, if they're eating, their serotonin levels might change. If they're getting mad at each other, then the adrenaline, epinephrine might be different. So it'd be interesting to be able to continuously monitor the neurotransmitters, but then correlate it directly to what we're seeing on the video. The other application of this that we worked with Martin Battelle a little bit on is looking at traumatic brain injury. So here you could use the same sort of box to look at different types of neurotransmitters. But the advantage here is that the patient doesn't move like the animal does, so it's much easier. You can just have it as a box. The disadvantage is it has to be approved by the FDA and multiple agencies before you can use it. But people have put microdialysis probes in traumatic brain injury patients and collected samples to analyze what's going on in the brain of these patients, especially related to energy metabolites like adenosine, catecholamines, and aspartate and glutamate and GABA as amino acid neurotransmitters. We'd also like to look at what's happening, since it's an oxidative environment, what's happening with oxidative stress. So that's another application, and we've had several people in the lab working on developing protocols for these different compounds. So these are some of the compounds that different people in the lab have worked on. So I guess what I want to show from this is that when I talk about a separations-based sensor, the idea is to be able to monitor multiple analytes simultaneously. So we can look at all the catecholamines. We can look at several amino acid neurotransmitters. We can look at multiple energy metabolites, adenosine, inosine. We can look at redox or reactive nitrogen oxygen species like nitrate, glutathione, ascorbate. We haven't done anything with neuropeptides yet, but we are working with Giuseppe in Italy on carnosine, which he's interested in looking at how that affects neurodegeneration and oxidative stress. So really the goal is to sort of have a modular platform where we have, excuse me, the probe that we need. So it can be here, mostly I've talked about brain applications, but it could be other applications, drug metabolism, or even bioreactor monitoring. And then we developed a specific separation for the compounds we're interested in. For some applications, we might have to do sample derivatization, like amino acids or glutathione. And then we can have integrated detection. So it can be electrochemical, but we've also developed some portable fluorescence detectors. And we're working on another sort of bipolar fluorescence chemiluminescence detector that we could use. So with that, I just hope I've showed you that I'm a big fan of microchip electrophoresis. I think it's really a useful method. It's fast separations of charged analytes. We're working on some potential clinical applications for monitoring oxidative stress and Alzheimer's disease. You can also, with the chips, integrate it into pumping systems and sample prep strategies to do single cell analysis and microdialysis sampling. And then portability is key for the on-animal monitoring and near patient monitoring. So we're still working on these nitrated peptide separations, and hopefully we'll move to mitochondrial proteins in white blood cell lysates in the future. Jay's been working on making the number of analytes that we're measuring with the single cell analysis system larger, and to include superoxide and some other RNOS. And then we'd also like to continue the online monitoring. So Jay is here. Indica just graduated, and he's been working a lot on the electrochemical detection. Danushka is just come back, but he did a lot of work on the tyrosine derivatives. I think Emily's here. I don't know if Kelsey's here. Kelsey, I think, is in one of these. Kelsey Shilley worked a lot on the peroxynitrite things. And then Anton and Galina and Rachel Saylor did a lot of the catecholamine work. So, and then I have to thank everyone here. I mean, there's a lot of people that have worked on this over the years, including Fracassi and Diogenes and Richard Pfeiffer, if some of you might know him. And then with that, the last thing I wanted to say is, if any of you want to come to visit Kansas, we're only about 30 minutes from Kansas City, and it's the host city for one of the host cities for the 2026 FIFA World Cup. So I was talking to Fracassi that maybe we need to have a bioanalytical symposium the week before the World Cup starts, so you can all come here and then go off and see the games. With that, I'd be happy to answer any questions.
Thank you, Susan, for the excellent presentation. Just a compliment, the best team in São Paulo is Corinthians.
Corinthians, yeah, okay, yeah.
A compliment.
I should have worn the jersey, right?
Just kidding.
I remember Richard was
São Paulo, I said in São Paulo.
Rio, right? I think Richard was big on Rio de Janeiro.
Any questions for Susan? Yes, one here.
Um, First of all, thanks for the talk. I'd like to ask if can this technology be used for early and widespread detection of neurodegenerative diseases? In other words, are there any economical barriers and or limits of detection, such as minimal concentration of the analyte?
Yeah, that's a really good question. So they have some preliminary data, but there's a lot of sample prep and enrichment that goes into the strategy to be able to get the nitrated peptides. Um, they are detectable, at least for in the Alzheimer's patients and the people that have neuro degenerative disease. Are you talking about just the blood or just in general? Um, I so um, I mean, I think the key is to have some sort of sample enrichment to enrich the nitrated peptides. We could do that with some sort of affinity chromatography. Um, uh, for the they right now they have to take quite a bit of blood to isolate um, the proteins and then measure the nitrated peptides. What we were thinking is if we could find specific proteins that result in specific peptides that were highly nitrated during oxidative stress, then we could sort of develop a unique chip just to look for maybe there's like one peptide or a family of peptides that show up in a lot of these disease states. Then we could use that as a strategy to measure the oxidative stress. Does that make sense?
I have two here.
Hi, Susan. This is Dozil speaking.
Oh, hi. How are you?
Can you hear me?
Yeah, yeah. Can't see you, but I can hear you.
I'm very interested to visit your lab in 2026.
Oh, please. Yeah.
to to hear and see you again, and I'm looking forward to see you in November in Lacey. But
Yes.
I'm curious about the electrochemical detection
Mm-hmm.
because you know that have some problems with electrofield decoupling and some alignment of the electrodes.
Right.
How do you solve this, or there is some room to improve this field?
So yeah, I sort of left a lot of that out of this talk. So there's three basic approaches. You can you can put it in the channel, you can put it at the end of the channel so the separation field doesn't affect it, or you can use a decoupler. The decoupler gets the best detection limits, but then you sacrifice some efficiency, separation efficiency. One of the approaches that we're looking at, and I talk a little bit about this when I'm at LACE, is we've been working on changing the electrochemical signal into a fluorescent signal using a bipolar electrode. So we bias, we get the electrochemical response. We use the bipolar electrode. One part of the electrode is an electrochemical detector for the microchip electrophoresis. The other part of the bipolar electrode is a detection electrode with either a fluorescent substrate that becomes fluorescent when it's oxidized, or a chemiluminescent substrate. So we've developed a system where we're sort of hoping to get better detection limits to eliminate some of the noise problems with electrochemical detection and background. So far, the detection limits are a little better, but not significantly better than what we're getting with EChem detection. And with reductive, you have oxygen interference. So we've been trying to use this for reductive so that we can. I think we are getting better limits of detection than we would get with direct reductive electrochemical detection. In terms of alignment, it's really application dependent. So in the on the sheep, we use isolated, electrically isolated potentiostat because it's the whole sheep is floating. So there we just put it in channel. But for some of the other applications, we put it like now it's called near end channel. So it's right, it's either just barely in or barely out, like the electrode's right at the end of the channel. So it really depends on the how efficient we need the separation, how much electroosmotic flow we have, and also the redox potential of the analyte. If it's an easy to oxidize or really easy to reduce analyte, then it's a lot easier to get good limits of detection.
I have one more question here.
Hi, Susan.
Hi.
I would like to say thank you for joining us in this school. So thank you very much for nice presentation and contribute a lot in our school. So I have just a small question.
Mhm.
Could you tell us about the effect of the flow rate and the size of the microdialysis sampling
Oh,
device?
how big the probe is, or so it's 200 well, you can get really, so if you do mice, you can get very small probes, but for the rats, they're usually 250 microns in diameter. So you can probe specific areas of the brain with these probes. So they're small enough so you can implant them in small regions of the brain to be specific, like the hippocampus or the striatum or whatever. But they're available in a large number of a wide variety of sizes. So and also molecular weight cutoffs. So you can actually there's several vendors, but you can buy probes with a fairly low molecular weight cutoff for small molecules, a little larger for peptides, and there's some that you can even use for large molecules like proteins. And then there's also another approach called push-pull perfusion, which is really similar to microdialysis, which basically does take sample out of the brain and then replaces it with buffer. But most of the probes are well, the probes that we use, because we're mostly doing in vivo microdialysis, are usually about 250 microns in diameter, and then they go in a guide cannula. For the sheep, they're much bigger. We actually did buy we haven't done any sheep brain surgery, but we did buy all the equipment for it. And for sheep and humans, the probes are much bigger because it has to go deeper into the brain. So in humans, when they do human microdialysis, they actually, so when somebody has a traumatic brain injury, a really, really awful one, I mean, really bad one, they actually take off the skull to relieve the pressure, and they put pressure sensors in there and also sensors for oxygen to monitor the brain. So people have put the microdialysis probe in with those two other probes in these patients that have really bad traumatic brain injury. And so in England and in Sweden, I think it's done quite a bit, actually. I don't know how much in the US, but they're larger because the human brain's a lot larger.
Thank you.
Any other question? See the faces here, they are preparing to go to the lunch now.
Oh yeah, no worries. Yeah. I
There is
hope
no
I
other
too far
question.
over.
So let's thank again. Susan, thank you very much for coming virtually here.
Yeah. Thank
We
you
catch
for
up soon, Lacey.
Yeah, and I'll see you at LACE in November. And yeah, remember 2026, Kansas City.
It's on Microchip, World Cup Microchip electrophoresis.
Yeah, to the World Cup Microchip Electrophoresis Conference in 2020 or 2026 at the KU.
So much.
All right, thank you.
We have just announced. Oh, sorry, we switch to Portuguese again.
Oh, no worries. Yeah. Do you want me to log off?
Lembrete aqui.
Olá, pessoal. Só lembrar de novo e reforçar quem não adquiriu a janta ainda ou gostaria de comparecer, a gente ainda tem vagas para hoje à noite, a partir das 19h30 na churrascaria. E quem quiser adquirir o ingresso, é falar comigo, está R$ 69,90, então a gente pode ainda ter, se vocês quiserem comprar e adquirir, então só falar com o pessoal da organização ou comigo diretamente. Obrigado. E a gente volta às 14h.
Obrigado. Obrigada, sorry. Yeah, ciao.